Okay, now we have find out the sequences of transcription factor binding sites. We know how to find that they are binding with the transcription factors, but what is important is the transcription factor binding sites are present in different numbers. They are in variable numbers, and they are present in different sites in the genome. So location, orientation with respect to each other and their number in the genome is called as grammatical rules. We characterize these by the degree at which they are prevalent and universal. Prevalent in a genome and universal in the genome, like different their prevalence in different organisms. They're prevalent in different organisms of different areas. Their universal presence and the effect of that prevalence in the quantitative expression and the quantitative differences of expression and transcription dynamics, and what is the variability from cell to cell. Regulatory architecture composition. Burst enhancers that are studied so far contains multiple regulatory elements, but exact contribution of each is largely unknown. The multiplicity identity and definitive TFBS are commonly referred to as composition of a regulatory sequence. Okay, I will give you an example of this regulatory architecture. You will get to know well about it from that one. Okay, now multiplicity. For some activators, transcription expression level increase monotonically with addition of multiplication. Okay, what is a monotonic increase of expression? If we have one site, yes, we add one transcription factor, the expression will be lower. If we have two sites, we add more number of transcription factors to that one, and the transcription and the expression level will increase. But to a certain level, when there are number of transcription factors and we added the number of transcription when there are a number of transcription factor binding sites and we added number of transcription factors to that one, a saturation condition occurs where all of the available transcription factors bind to the all of the available transcription factor binding sites. The expression will get like this. It gets steady. For example, 13 transcription factors that were recently examined in yeast and 5 or 12 liver-specific transcription factors in mice and in human hepatocellular G2 cells. Now, what is it? Next is the affinity. Affinity of TFBS to their transcription factors is a sensor for concentration of transcription factors. What is it? If there is a lower affinity for certain transcription factor binding site to their respective transcription factor, expression will be lower and there will be no expression. But when we increase the amount of transcription factor, there are more chances of binding of these transcription factors to the lower affinity site. The expression will enhance. Identity. Okay, effect of identity of the binding transcription factor is also very important. What is an identity? Okay, Identity is the presence, is the appearance of transcription factor binding sites. How they come in the 3D structure, and their appearance leads to the binding to different transcription factors. So if the respective transcription factor comes good in the shape, like according to which model it was. Okay, if it comes fit in the 3D model, it will bind to that one. So this is called identity. Heterotypic clustering is there are different number of transcription factor binding sites for different number of transcription factors. Like transcription factor binding sites are in multiple, and transcription factor enzymes are also in multiple. It is heterotypic clustering. But when there are different transcription factor binding sites for the same transcription factors, it is homotypic clustering. So heterotypic clustering results in more expression than the homotypic clustering. Okay, this was the example I told you I will tell you later. This is the constrained architectures. We know that now we know that what are transcription factors, what are their binding sites, how to find them, how to find their binding, how to find their affinity, what is the effect of their affinity on expression. Okay, now there are in one for a single gene there may be more than one transcription factor is involved, and how these different transcription factors come together and what 3D structure they form is very crucial. So how the different transcription factor binding sites are arranged, how the transcription factor bind on them, and its sensitivity for the prospective regulatory sequence is very crucial. So there are two models for this one. Billboard model. Expression is robust to manipulations to transcription factor binding site spacing and order. Like if there will be more spacing or less spacing, relative spacing, the change in the relative spacing will cause the change in transcription. This one. Look at this one. There are three different transcription factors shown in green, pink, and dark pink. So there is the relative positioning and spacing of these three. How it decreases and increases the change, the expression. So if the pink one is in the mid, and green is on left, and dark pink is on right, so the expression is higher. If there is an now come for this one. If the green one is in the mid, the expression is higher. But if the relative spacing of this green one changes with this one, like this one, there is a space in between them, there is no space in between them, it causes a change in expression. This is that one example, enhanceosome. So what are the enhanceosomes? Enhanceosomes is a complex structure when different transcription factors are involved. For example, interferon beta enhanceosome. There are different transcription factors involved for this one: ADF2, C-Jun, IRF7B, IRF3A, 3C, CD, and NF-kappa B. These are all different number of transcription factors, and they bind to their respective transcription factor binding sites in the DNA. Yes, and they form a complex structure. Transcription will start only when this complex is formed. This complex is called as enhanceosome. Look, if the complex is not formed correctly, there will be no expression. What I explained today was from a paper of Michael Lebo and Aaron Siegel. Here is a link of that paper. You can find out many other things that I've skipped. Thank you so much. If there is any question, I can shoot you. Thank you.